We're going to start with the gene editing side of things. So I'll pose a variant on the first question up there, which is, for research purposes, what QC assays do people think we need to do of genome-edited iPS cells? Very broad one. So I think we can probably all agree that we need to look at, make sure that mutation is there or our gene is knocked out. I think that's a given. But what about off-target effects?
It needs to be holistic for all the lab involved. So basically, if you propose genome-wide analysis, that's great, but that's probably a really few number of labs that can perform that efficiently with the right pipeline of analysis and so on. So no, you need to provide something that is more kind of accessible in a way. So again, you can sequence all the sites that could be targeted by the guide RNA, which is one way. You can increase the number of cell lines using two sets of guides. The trouble of that is that, of course, you want to avoid to increase the number of lines, again, to reach a level that is not practical for anybody to use, because 12 lines, the number I've mentioned, can't be achieved very easily in a lot of labs. So you need to find a balance between all that. And for us, at least, using two sets of guides just look the easiest functional way to do things.
To reinforce the point that Ludovic made also during his talk is the way that one initially selects the guide RNA is actually really critical. So that aspect of it shouldn't be underestimated because it will significantly reduce the potential for off-target editing. In addition to sequence homology, one can use these experimental-based methods such as Circle-seq or digenome-seq or guide-seq, which are all kind of based on the same principle of just experimentally determine sites of double-stranded breaks, either in primary cells or in whole cultures. But sorry. But obviously, one of the limitations is it's not kind of at the single cell level, so it would be a bulk population, and one could miss something because of the depth that one sequence is at to interrogate. So. Yeah.
No, also, I think we discussed that during the breaks, that off-target effect is a problem, but is it the biggest problem?
Yeah. So in my mind, we know that large structural variants are relatively common, and they can emerge in culture. And if you are definitely going through these clonal bottlenecks during gene editing, might it make more sense to first look for, do you have an extra copy of chromosome 12, or do you have a chromosome 20 CMV? That may actually have a large phenotypic effect, as opposed to some off-target that may be non-coding.
And I guess it comes back to some of the points that I made earlier about the difference between looking at molecular level and actually what you're seeing functionally in cells.
It seems to me that the conditional target, like bringing in LOXP sites or similar approaches, is actually, maybe more telling and more straightforward, because there's clonal drift. So even if we generate a clone and, by targeting and we sequence it, maybe 10 passages later, P53 or another mutation actually expands within the population. Maybe it happens during differentiation and so on. But if we have a conditional allele, we can do this, we can knock out the gene now or in 10 passages from now, or, during differentiation or after differentiation and so on. So it's clearly more work to generate a conditional allele, but then again, phenotyping 12 lines is also not always fun. So, it's a trade-off, and I think by conditional alleles, you can actually do this in thousands of cells in parallel, right? Because you have the whole population that you knock out. And it's more time controlled in addition. So isn't that the obvious solution if we manage to generate those alleles?
Yeah, I think it's a great point, and I think that as technology evolves, and I think that's probably one of the points we're going to be exploring as we continue the discussion, as technology evolves, is there changes and new things that are coming that will allow us to get around some of these issues, and the use of conditional alleles is definitely one of them. What about when we're generating gene-edited lines? There was lots of discussion earlier about doing testing for pluripotency when we make iPS lines, or looking at genetic stability. Should we be doing that after we gene edit lines as well? Or is that over the top? Is that realistic?
I think genetic stability is...
I think if we're doing a good job growing our lines and they started off as pluripotent, I think they're likely to stay that way. But in terms of looking at has there been something large and unwanted that is likely to have a phenotypic consequence that has either emerged from the population because we've gone through this clonal bottleneck or emerged during gene editing, that's something that I think would be good to look for. But in terms of what level of resolution one looks at this, that's another topic for discussion.
Thank you. I think another topic that slightly complicates this is the mosaicism of the parental line, which is usually not really regarded. So if you start with genome engineering experiment, and nowadays you have a parental line, and then you have a genome engineering experiment, you pick a clone or two clones, and then you compare those two clones with a parental line.
So just to answer to that, no, we use untargeted, what we call untargeted lines, which is incredibly, but it's happened with CRISPR, sometimes can be difficult to get because CRISPR is so efficient. But the total untargeted line, you can then see the reverse. That's a cell line that goes to a CRISPR event that is not targeted. And then you can also become a bit nervous because why this cell line is not being targeted, despite the fact that we see the same dose of CRISPR, the same good dose of guide RNA. So I agree it's a key challenge, but that's what we advise right now is to have an untargeted control.
So as I mentioned, we in our lab we adopted the same approach as Ludovic have taken untargeted lines and using them as controls. And anecdotally, we do see differences sometimes between the untargeted lines compared to the original wild type that their assay would have been set up. So we're trying to differentiate lines into neurons, for example. The wild type might work brilliantly well, but when we take out untargeted cells, all of a sudden actually that assay and differentiation changes quite a lot. So it is a very good point and a challenge, I think, absolutely, for the field. Has anyone got more comments on that side of things? Or pose a slightly question, what about clinical lines? That changes things, presumably.
I think what Martin brought up is a very serious issue, which I hadn't thought about until Martin brought it up, which is actually we are testing for pluripotency of the therapeutic product right at the end. But I mean, what Martin just presented is why we're doing that, because actually something may mutate further down that manufacturing, which is nothing to do with your pluripotent cell. It's a cell which is differentiated and is changing its genetic makeup. So can you give us anything better than we're doing at the moment? And the answer seems to be not really yet, which is fine, okay. Means you need to do more research. And then the second thing is, collecting that data is going to be very important for the future evaluation of whether a therapeutic works or doesn't work. So is there anything there in which you can help us in terms of the genetic stability? If you get a change in chromosome 12 or 20 or 17 or whatever, does that make that cell less efficacious? And obviously, we're looking at that as well. So clinically, that's what we're concerned about. Not whether you can necessarily do a lot of testing on your pluripotent cells, but whether that therapeutic cell is safe and whether it's going to be efficacious. And therefore, what in your standards can help us reassure ourselves that that's going to be the case.
So that's kind of along the lines of what we're doing in the UK Regenerative Medicine Platform 2, essentially taking some of our common genetic variants and P53 mutants, differentiated to dopaminergic neurons and some other types of cells, and looking, can they differentiate efficiently, and if they differentiate, are they still good functional cells, and ultimately what they do in vivo. So we are kind of trying to cover those exact questions.
One of the things actually we need to be careful of when it comes to the chromosomal gains is that we can happily talk about gain of chromosome 17, for example, but actually it's never the whole of chromosome 17. It's mostly bits of the long arm, and you can have several lines, all of which have got a gain of a bit of 17, but they may be overlapping, but there are other things. So the assumption is that somewhere in there, there's a gene that is giving the selective advantage of the stem cell, but there's a lot of other genes being carried along, and they may have a consequence for the differentiated cells, but actually different lengths of it may have different genes. So you may have a short bit of chromosome 17, doesn't have a nasty gene in it. A bit longer might have a nasty gene in it for the consequence. So you've got to think about what actually is the variant when we're looking at karyotypic changes. It's not that just they're not all the same.
There's a difference between culture-induced variants or mutations and spontaneous ones that are quite fine. Have we looked at that or distinguished them? Can you see a difference between culture-induced variants?
Yeah, for sure.
Versus
I mean,
in the general population in bone marrow, for example, is what you said. Are they typical culture-induced?
So I think there are distinct differences between the processes that drive mutation in culture and in the skin, for example. In the sequence patterns that get mutated in skin are very different and very characteristic. In terms of consequences, I don't know that, for example, UV produces more deleterious mutations than something else. But I think the message from our work is that they are more likely, in general, to be deleterious than the germline. So things that occur both in culture and in the somatic tissue are more likely to be deleterious, not necessarily because they confer a growth advantage, just because there isn't as much selection. One thing you can use to prioritize mutations is to look at population genetic resources. And the reasoning there is that most of the time, mutations that are deleterious, bad for you in some way, are just rarer. They're rarer in the population. And you can, the ability to detect those scales with the size of the resources. And we have really, really big resources in human genetics now where we can say, well, we've looked at 100,000 people and we have never seen a knockout of that gene in healthy people. So you can't make an exact comparison because you'll never see really the same mutation twice. But if you say, well, that gene is, we never see an LOF, and we see it in culture, you can begin to maybe extrapolate. If it's not found in healthy people at some frequency, then maybe we shouldn't put that into people. But again, it's not an exact science.
In our own work, we also see that the overall mutational burden that you see in these human embryonic stem cells is very similar to what you see in people. So there isn't I think that tells us that there isn't this excessive burden beyond the huge amount of variation that you see from individual to individual. But if you restrict yourself to analyzing those mutations that either are not seen or that are very rare, and that's what we do, and a small fraction of those are in ClinVar, and we actually know what the consequence is. Most of them are not, but you can predict which ones are likely to be highly deleterious. And then you look at which genes or gene classes are seen recurrently, even after you've hit all of those kind of stringent thresholds. Then you can start to maybe get some sense of which ones are playing some functional role in the context of stem cell culture.
And I think,
but that's a small subset, and we still don't know functionally what is this going to do to differentiation. Does this make the cell unsafe to put into a person?
No.
I do not have the answers to those questions.
No. Actually, it's probably more informative to look at cancer studies, cosmic cancer gene census, and cancer genome atlas, and so on. That information is accreting all the time, and we will be able to be better able to predict consequences of loss of functions in certain genes. But it's never going to be, that will have that consequence in that differentiated lineage.
Just to go back to the question you asked originally, which is genome editing in the context of clinical applications. And so what occurred to me is the comment you made about a targeted control versus a wild type unedited control being different in your experience. It made me think about a couple of papers that have come out recently, one from Alan Bradley, Dieter Egley, Maria Jason, and others, which have warned about these larger deletions that would go undetected using what would now be kind of a more standard method that's used worldwide, which is just targeted deep sequencing using amplicons of about 300 to 400 base pairs, and mutagenizing any one of the PCR primer binding sites would make one believe that they have a wild type allele at that on-target site, not realizing that they might have completely kind of deleted a huge fragment of a chromosome around the cut site. So that's a word of warning, I guess. And in clinical context, it becomes much more important to evaluate that possibility. And it's not a standard protocol.
Yeah, I think it quite nicely, I guess, illustrates the importance of understanding the limitation of whatever genotyping strategy or assay and things that you use and what it might actually possibly be telling you.
Yeah, and that's where I think that these whole genome sequencing approaches coupled with the CRISPR-Cas9 technology in a clinical context will be incredibly important, ideally at the single cell level.
I think I would also add that this study and many others have looked at technologies where CRISPR-Cas9 is active at high levels or present for long periods of time and is just cutting and cutting and cutting again. And I think what we don't have a very good sense of is to what extent does that raise the possibility of these larger deletions. And if you use a very rapid kind of hit and run RNP transfection, is that going to be as big of a problem? I think it is going to exist, but is that something that we can minimize if we don't try to get really super efficient gene editing?
So I just take this back to the clinical scenario for a moment, and just that there's never going to be an absolute risk associated with any of these changes, that it's always going to be interpretative. If you're going to be altering a life-ending disorder, you might accept a higher level of risk. So I think it's not necessarily being able to make that decision on the cell line, but to provide the information that the clinician can then make that risk-benefit, and the regulator, more importantly. So I think that's where we're going to trip up. This may be sacrilegious in this room, but the pluripotent stem cells is a starting material. It's not the be-all and the end-all, and I think the move, as Martin was saying, to be testing the final product is so much more relevant and so much more realistic, rather than getting hung up on what the PSC line looks like.
To try and sort of finish up with the gene editing piece, I guess it comes back to a point that was made earlier about should we just be doing straight knockouts or straight edits, or should we be using other technology like conditionals to try and minimise the risks or minimise other effects? Is there technology out there that people are aware of coming through that actually could impact on this, or reduce the number of double-stranded breaks, for example, to impact on cells?
It won't solve all these problems, but multiple guides for your gene, multiple genetic backgrounds, it doesn't have to be hundreds. Even one more than the one you're doing will add a lot, I think, in terms of robustness of results. I know that's kind of an obvious point, but I don't want to, it might be, it's easy to think of it, we could solve it by sequencing or whatever, but often if you see the result twice in two different genetic backgrounds with two different guide RNAs, that's already a lot stronger than just the one line.
So I don't know if anyone's got any thoughts on the new technologies and side of things.
I don't know if anybody has experience with base editors yet. I think somebody during the lunch break mentioned them, but that's a methodology where it doesn't require the induction of a double-strand break. It has its limitations, and it's still not widely used, and I think the efficiency is not quite as high as standard pyogenes Cas9 based editing.
Base editing is definitely less efficient than CRISPR because you have multiple levels of components that need to act. So you just multiply probabilities. So it's just pure maths. For in vivo work, base editing will definitely be the future, because the problem with indels is that those knockouts are created by reading frames, by changing the reading frame. So you are always creating a truncated protein that does not exist in nature, which is also called an antigen. So you could develop immune reaction against a truncated protein that you are creating by generating this knockout. And you don't know what is going to happen because those indels are completely random. So base editing will definitely be the future, to engineer specific stop codons to make that knockout. So then you just pick up like a tryptophan which is the GAG, and then you just transform into a TAG, which is a stop codon, and there is no way you are creating a truncated protein. So base editing, you'll start hearing more from it from an in vivo perspective for knockouts in the future, mainly because of this fact that you are not creating strange proteins that the immune system is not used to.
Just to mention, it comes at the price, of course, that you put a base editor into a nucleus hooked up to Cas9, which will be scanning the entire chromosome, bind to every single pump site, and do stuff in the vicinity. The beauty about the double-strand break Cas9 is that it will only actually do the double-strand break if it's a perfect or near-perfect match across the whole 20 base pairs of the guide. That's not true for base editors. That will just be active as they scan along.
Has deposited a few plasmids in AdGene that are an improvement over the original versions of the base editors that are supposed to make that probability a lot less. So it's worth looking into.
Okay, do you guys want to go with the genetic stability?
Sure. So I guess part of the question for this group, I mean, this is a fairly aware group of investigators, but I wonder what the general impression is of quality control standards in this field, publishing nowadays.
I was trying to publish a paper on just the validity of different methods for detection, and one of the reviewers, who was obviously from a cytogenetics field, it seemed to me, said, well, this is all really well known in the cytogenetics field and there's nothing novel about it. But my feeling was that in the stem cell field, people are not as well informed, or were not at the time. So I just kind of took a number of papers that were recently published in relatively high impact factor journals, and on a small sample that I've analyzed, only about 50% of them even mentioned anything about the genetic status like karyotyping of the cells. And of those that did mention, again, only a very small fraction, maybe 10%, actually described how karyotyping was done. And if you really understand the karyotyping, it actually affects, or the results, the sensitivity, depends on the number of metaphases that you analyze. So, whether you analyze 20 or 30, well, the interpretation is different. So my feeling is that, people don't pay too much attention to this at all, and should. At least to flag up, just to flag up for the future, what the cells actually contained or did not contain.
No, because we've seen today that there is a massive number of assays you can do that can be extremely complex and detailed, like genome-wide sequencing, to a more kind of qPCR-based test. No? So, I mean, you need to define the bar of those assays. And I don't know, but no, I think the classical way we have been dealing with that so far have been karyotyping. That's the historical way we've been dealing with things.
Beyond just looking at what was done to assess the genomic stability of the cells. So we tried to, when we were writing up our story, we tried to determine, okay, what was the culture history of these cells? Under what culture conditions did these mutations arise? And this turned out to be very difficult to do. I mean, a lot of the papers just simply didn't say how they grew the cells, what passage they were, where they got them from. And I think that's a real lost opportunity to try to understand. It would have been very helpful, I think, to be able to trace back what the life history of those cell lines were.
Can I just make a general point about karyotyping, and before that becomes something that everybody agrees is a good idea? There was a comment earlier about it, the cheap and cheerful SNP arrays, and I think that karyotyping is not really carried out by most of our diagnostic genetics laboratories anymore. They do tend to use SNP arrays, and karyotyping is, it's considered more old technology, and so I think it would be a shame to tie ourselves to that as our standard baseline test. I think SNP arrays achieve as a similar, initial look at chromosome changes.
Freeze thawing issue, which you raised, which is really interesting. I just wondered if any of you have actually looked at that at all.
So actually mixtures of cells from different individuals change with freeze-thaw and change with passage. I mean, as you would expect, it's very substantial in terms of lines that compete with each other. There's lots of, so if there's a mosaic line, fraction of cells carrying that mutation, it wobbles enormously from passage to passage and from freeze-thaw to freeze-thaw. And it doesn't, at this early stage, seem to have any kind of, you can't say whether it's going to go one way or another. It's just random, but it has a huge effect.
So maybe just to add to that, Richard Davis, who's here from the lab, he was curious what happens when you actually freeze down differentiated cells. What happens to their phenotype, not so much their genotype, because many people put cells into patients that have been frozen thawed, or we buy commercial lines frozen thawed. And there seemed to, if I'm summarising right, no difference at least on the electrical properties of the cardiomyocytes, so that was encouraging.
I've listened to a lot of your sort of, I'm a nature editor, and what type of characterization you need, a smaller scale study or something that people, I don't know, this freeze-thaw characterization, for instance, is that publishable? What do you do when you've got your, freeze-thaw characterization results? Are we concerned about sharing these, what we call so-called insignificant results? Shouldn't we, preprint servers are the rage at the moment. Shouldn't that be something we, as a community, we should all be doing?
In our case, we'll put that in a bigger paper that has a lot of other stuff in it. It was part of I don't know if we ever thought about that as a paper by itself. It's very hard to predict those things. You just think this is something we should do as part of a wider study. The results are interesting, and it will go in a publication somewhere. But you're right, it's hard to publish that very, standards-based quantification, community resource thing. In bioinformatics, for example, you get quite papers that are, maybe even not published, for example. There's a famous example, BWA, which is one of the aligners, that the lead author got so tired of being reviewed and so on and so forth, he just said, fine, and left it. It's now cited like 3,000 times. But it's hard to, sometimes it's hard to convince those things that are of enormous use to a community to get them out there.
Yeah, I had a lesson in this when we started the whole issue of genetic variants in ES cells. When I started talking about, we found these variants in human ES cells. And several people working with the mouse stood up and said, oh, we see that in the mouse too. And I said, oh, that's interesting. Where is it published? And which ones? Almost nothing was published about it. It was known it was sort of folklore somehow in the community. But I think we found one paper.
There is a general comment on that. That's Daniel and Florian doing their stuff in their lab. And that may not be what I do, or Peter does, and this kind of things. And that's the problem to publish this kind of paper. It's an observation of a specific lab using a specific technique. And so if you want to, and that's the whole problem of all those systematic studies, you want systematic studies using multiple lines, multiple labs, multiple techniques before you reach any conclusion, because otherwise it's just occasional observation.
This sort of thought which Martin touched on was this thought we have for an ISCI study to try and create a database of all the sort of variants that exist out there, what happens with them in different experiments, where people can put information in that we can kind of collate this kind of information so that we can have a potential for looking back over the coming years and say whether any of these things mattered or not. That's...
Yeah, I think one thing I was going to ask some of the group is, well, everyone knows we kind of know what genetic changes cause human cancer now, and we have a vast amount of information on human developmental genes. But is there an easy way for people to access this information and get at it? How simple is it?
The easy end is the list of cancer genes, of which there's about 500, right? Yeah. Structural rearrangements, a little bit harder again. The kind of population genetic resource, again, a little bit more difficult. But all of those things are reasonably accessible now. It's more that people just need to know they exist rather than it's really difficult to work with them. I think it's just, that was an exercise for us in going through, right now we've got all these mutations, how do we classify them? And there's no one way to do it. You just have to try and dig for as much information as possible.
Sure, sure.
Florian, you probably have a...
No, I just wanted to say that I think this is exactly the right approach, that we're, even with 100, 200 cells, we're still underpowered to see a lot of things. And so the more that we can pool together what information is out there into a centralized resource, I think the better chance we have of understanding the underlying biology. So I think it's exactly the right approach.
But also I think there needs to be some quality control around that analysis. So if
Sure.
everybody's doing something their own way, and if we had access to a resource where they were listed even, we knew if you wanted to get a SNP array done, you could get it done easily and get a validated result, or where you could get whole genome, whole exome seq done with some interpretation regarding to cosmic, for example. That, I think, then adds great value to the data, and that, to me, at the moment, seems to be a bit of a gap.
Yeah.
And I would say, I think the experience in human genetics is those sorts of problems have only really been solved, the consensus and variant calling and so on have only been solved by large international consortia where everybody pitches in. I mean, 1,000 Genomes is the perfect example. Before, we didn't really know how to call variants from short-read data, and afterwards, however imperfect, we had some sense of what works and what doesn't. I think they get a lot of stick, actually, and I understand because they suck up a lot of money, but they do produce really valuable things like variant call formats and so on that the entire community uses. So I don't think we should undervalue them.